So as we look at the replication bubble, when we're copying DNA, we've got the helicase enzyme shown in blue here, which is breaking the hydrogen bonds, which are holding the two strands together. We've got the single-stranded binding proteins, which are shown here kind of in aqua, that are helping to keep the two strands propped open so that they don't reconnect or reanneal with each other. And then we also had an enzyme called Polymerase that came in here and actually synthesized or made an RNA primer directly on top of the DNA template strands. Now this RNA primer, I do want to emphasize that those bases, they are connected to the template DNA. So they still have hydrogen bonds between them. So it is a specific interaction that those have with the DNA strand. The point of the primer is that the primer is going to really provide the required 3' end. And that's the required 3' end that DNA polymerase is going to need. So once we have that end available, DNA polymerase is going to be able to come in here. So it is kind of a large enzyme. So I'm going to just draw it like this. But DNA polymerase is going to come in here and it will start to read the template. So as it does this, it will look here and it will see a T, and it knows that across from a T should be an A. So it is going to add that A onto this new strand. And we're going to connect the DNA, which I'm showing in green here, directly onto the 3' end of the RNA molecule. And once we've done that, and that A will hydrogen bond to the T that it's across from, we will look at the next nucleotide, which happens to be a C in this case, and we know that across from that goes a G, and we'll look at the next one and we'll put a C, and so on. So as we continue to add these on, notice that the 3' end of this strand is getting a little bit bigger. So it's getting longer than it was before. And we would have the same thing happening down here. Across from a G would go a C, and then we've got a G and an A, and so on. So the 3' end is getting longer. So DNA polymerase is going to continue to do this, and notice in this example that it's kind of run out of empty nucleotides to look at. And so this is where this process is just kind of a continuous process. And as DNA polymerase starts to move in this direction, so if we just draw our directions on there that it's moving, so DNA polymerase is going this way, and we see up here that it's going this way. As it does that, the helicase enzyme is going to actually move forward with its job. So we will see that helicase actually starts to break more bonds. And so we're separating the strands even more, and we're making the replication bubble even bigger than what it was before. So our replication forks are going to move. And they are moving, in this case, since we're talking about the bubble and there's two forks, they're actually moving in opposite directions from each other. So if we draw kind of the new picture that we're going to have, okay, so here now I have altered my picture to show that the replication forks have moved apart, and this is the result, first of all, of the helicase continuing to break hydrogen bonds, and those single-stranded binding proteins are going to be sliding back along with the helicase as well. Now, once we have more DNA here that the DNA polymerase can actually see, it is going to continue to add on to the 3' end that it was working with. So it will again continue to look at the old DNA or the template strand shown in black and incorporate the correct bases, nucleotides across from it. And we are linking each one of these together. So don't miss the part that we are forming phosphodiester bonds as we link these. So across from the C would go a G, and so on. So this would be my new 3' end right here. And we would be doing the same thing down here on this strand. And there's my new 3' end down on that place. So what we've done here, if we just add a little bit of detail, the green is what we would call the complementary strand. Okay, so this is going to be complementary in that it is the opposite of the strand that we're adding it to. So in other words, where there's a T, there's going to be an A across, and so on. And we could also call this the new DNA. Obviously, this is what we're building here. And notice again that it's really new DNA that's connected to a tiny little piece of RNA here shown in the purple or the pink color that we have. Now we do run into a little bit of a problem here. As you'll notice, we've got this gap right here and we've got this gap right here, which we have not started to copy. And the reason for that is that the DNA polymerase cannot or can't add to a 5' end. And notice that is exactly what we have right there in our primers. So those are 5' ends, and the DNA polymerase really doesn't know what to do with that. So we have to have a way of solving this little problem, and what's going to happen is that we're really going to have another primer that's going to have to be synthesized and added to the DNA strand. Okay, so primase is going to back up as far as it can in the replication fork, and I've drawn this rather small with the number of nucleotides that I put in here, but it would back up as much as possible and it would look at what's there and it would start to build the primer right on top of the DNA as it did before. So across from a G would go a C, and across from a C would go a G, and I'm gonna make it just that short right there. But again, the primers are always about 8 to 10 nucleotides long, and it would be primase that's doing this, and it would build it anti-parallel to the template. Okay, so I'm gonna write that one right there. I would have another one over here. So if I just write this one in, so we've got G, A, and T. And again, this would be my 3' end, and this would be my 5' end back there. Once we have that, of course, DNA polymerase can see those ends that are available, and it can actually start building DNA again. So it's going to come in here and it's going to add on to this 3' end. So across from the C would go a G. So if we just start drawing that in, here I've got my G, and then we'd have an A and a G and then a C. Okay, and now we've actually, so that's my 3' end. We're actually running into this primer, this RNA piece that's stuck on the DNA. And we do want to get rid of that because we want a continuous piece of DNA. We will have a different DNA polymerase that is actually able to come in here and remove the primer. So we will have DNA polymerase remove the primer. And so I like to think of this as it's just kind of scraping it directly off of the template DNA. So as it removes that primer, and so if I just get rid of it here, now we've got more DNA that we can write directly on top of. So it's going to continue to extend this 3' end. So again, using our base pairing rules, and this is what we would end up with. And so now at this point, this is my 3' end right here, and this was a 5' end that we had that was connected to that primer. We do have a little problem here because DNA polymerase, another thing that it cannot do is it cannot make the final connection. Or in other words, it can't make the final phosphodiester bond. Okay, so since it cannot make this final link, the DNA polymerase itself, we will have to have yet another enzyme that comes in and actually makes that last connection. So the enzyme that's going to be responsible for doing that is DNA ligase. DNA ligase's job is to connect the final nucleotides to produce a continuous strand. And so since it is connecting nucleotides, it would be a phosphodiester bond that is forming. So it will be able to come in here and it's going to just make that last little connection between that 3' hydroxyl group of 1 and the 5' phosphate of the next one. And so at that point, what we've now got on that strand, if we just draw it kind of on the other side here, just to show a little more detail, I've got a 5' end, and that 5' end is connected to DNA. So that 5' pink, that's RNA, It's connected to DNA. It's continuous, and it runs all the way up to here, and that's my 3' end. So that's what I've made. And we would have the same thing happening up on the top. We're going to have DNA polymerase is going to be able to come in here, and it's going to add on to the 3' end of the primer. And so across from the T would go an A, and so on here. And we would be removing, scraping off the primer as we go along. So if I show that, and we continue to make the DNA using our base pairing rules, we're going to need ligase yet again to connect that last connection. So here's that last connection, and I'm just showing it in red to indicate where it's at. But we would then have our continuous piece where we start off with a little piece of RNA, and then we have more, we have DNA extended from that, and it would be the 3' end. Now, about the way that I've drawn this, I always draw a little arrow on the 3' end like this, because the 3' end is growing. That's the end that's always going to be getting longer. So if you put the little arrow there on the 3', that'll help you to remember that that's the end that we're going to keep extending and getting longer and longer all along.